I hope Miss Alia Fardous is left. Next participant, Jitender Singh Pachaya. Next participant, Dr. Jitender Singh Pachaya. Next, Dr. T. Lavanya. Dr. T. Lavanya. Good afternoon. Good afternoon. No, no, no. No, sir, not visible. No. So you want to make a time? Sir, you want to take a time? Then I call next participant. Thank you. Next participant is Chahat Sarma. Next participant, Chahat Sarma.
Good afternoon, everyone. Am I audible?
Yes, yes, audible.
Actually, I have sent my posture to Karishma ma'am. Please share it on the screen. So, good afternoon, everyone. Myself, Chahat Sharma. I'm a PhD scholar from MMDU, and I'm doing my PhD under the supervision of Dr. Pooja Sharma. And today I'm here to present my topic on the poster named A Biological Approach to Degrade the Cypermethrin Accumulated in Soil. So cypermethrin is basically insecticide which belong to pyrethroid class of pesticide, and it is the maximum used pesticide in the fields as well as in households and veterinary areas to kill insects, mosquitoes, flies, ticks and mites, etc. If we talk about on the worldwide level, then around 3 million tons of pesticide is being used worldwide, out of which 2.5 kg per hectare is being used in India. And from that, 90% of cypermethrin is used in the fields of cotton, basically in Punjab and Haryana fields. And it has been seen that from some times, the level of cypermethrin has been increased in soil due to its accumulation, and this cypermethrin is causing so many damaging effects. These damaging effects include environmental pollution, health issues, pest resistance, as well as bioaccumulation. And if we talk about the fate of cypermethrin in environment, then it is persisting in soil, and then it is persisting in water through rain, as well as through drift and runoff, as well as it is deposited in the form of some of its sections deposited in soil after its decay of half-life. So to those particular points of cypermethrin which are present in soil, they are causing harm to the environment. And to degrade those pesticides residues, there are several methods, physical, chemical, but all those methods are not environment friendly, as well as they are costly, and they take time to degrade the pesticide. So we are looking for the biological approach to degrade those accumulated pesticide in the soil. So we thought to use nitrogen fixing bacteria. These are the very crucial member of the soil and used for the crop production. These we use those bacteria to degrade cypermethrin as well as many other pesticides from the soil which is accumulated after their use. So we have diagrammatically shown here our procedure, biological approach to degrade accumulated cypermethrin from the soil. So here what we have to do is we have some isolates of nitrogen fixing bacteria in our lab. We use those bacteria to grow on minimal salt media, which is having some concentrations, different concentrations of pesticides. By using those different concentrations of pesticides, we inoculate our bacteria on the minimal salt media, and we check the tolerance power of bacteria in the presence of pesticide. If our bacteria are able to grow in the presence of pesticide, it may show they have some capacity to degrade them. So from those petri plates having minimal media as well as pesticide, we isolate the pure colonies of bacteria which is tolerant to pesticide, and then we do further studies to check their ability to degrade the bacteria on the basis of their morphological tests, physiochemical, biochemical tests, as well as some confirmatory tests using HPLC GCMS. And from that, we particularly choose those stochastic nitrogen fixing bacteria having dual activity. One is nitrogen fixing and other is pesticide degrading activity. So this is the basic approach to degrade cypermethrin from the soil. Thank you.
Thank you so much, ma'am.
Thank you, sir.
Miss Alia Firdaus.
Yes, sir.
May you want to present again?
Yes, sir. Yes, sir.
Please proceed.
Is this visible?
Yes, visible.
Okay, sir.
Just
So,
zoom out.
zoom. No, okay.
Please zoom the meters.
Is this okay?
Okay.
Okay. So, myself, Alia Firdaus, and I am a PhD scholar of Integral University. I am doing my PhD under the supervision of Dr. Uzma Afa. So, the topic of my poster presentation is the pesticidal potential of wheat plants against some common phytopathogens. Sir, am I audible?
Yes, yes, audible.
So, as we all know that agriculture pest management is a big concern nowadays and also from last many years. So, as compared to other plant parasites, fungi is the main cause which affect our disease and also the crop production losses. So behind being harmless and non-phytotoxic, it has been proved that plant extract exhibit inhibitory effect on pathogens. The plant-based pesticides are cheap, locally available, non-toxic, and easily biodegradable. Using biopesticides and biofertilizers together, which enhances the soil health, reduce environmental pollution, and support sustainable agriculture. Basically, the active principle of this biopesticide is in its secondary metabolites. Secondary metabolites include the alkaloids, tannins, flavonoids, phenols, which have the potential to kill the phytopathogens and also have the ability of antibacterial, antifungal, herbicidal, and insecticidal actions. Basically, the aim of this study is the collection, identification, and phytochemical screening of the wheat plants. Wheat plants are basically those plants which are growing anywhere where it is not wanted to grow. And then the extraction and characterization of the important bioactive compound from the selected wheat plants, then evaluation of the pesticidal potential of those extracted bioactive compound against the phytopathogens which affect are the crops. In some literature review, different work has been mentioned on wheat plants and also proved that to be effective against the different phytopathogens. In some literature, Parthenium histophares are well written, and apart from Parthenium, one more plant is there, which is Agaricum conizoid, which is highly effective against the species of Alternaria. Lantana camara is one of the wheat plant which is effective against the Ralstonia solancearum. This bacteria basically affect the brinjal plant. And apart from these plants, modern investigation have supported the claims of the insecticidal activity of several plants like Allium, Sativum, Artemisia, and different plants. So this study is basically to explore the biopesticide for important phytopathogens. This table shows the list of different wheat plants which have the activity against the fungus which affect our crop. Lastly, concluding by saying that as we know that wheat plants are those plants which can grow anywhere where it is not wanted to grow. So behind vanishing or destroying them we can use them as pesticide and as some wheat plants have already well mentioned activity of pesticides. So proposed research will have the solution for the management of phytopathogens along with the controlling of population of other pests. These are the some references, and thank you.
Thank you so much, ma'am. Nice presentation.
Thank you, sir.
Now I would like to invite Ms. Anu Serawat. Ms. Anu Serawat. Ms. Anu Serawat. Ms. Anu Serawat, you are not audible. Dr. T. Lavanya, are you ready? Dr. T. Lavanya, are you ready? Dr. T. Lavanya. Mr. Pradeep Kumar. Mr.
Hello.
Pradeep Kumar.
Yes.
Yes, sir. Sir,
Hello.
are you ready?
Yes.
Yes, sir. Please share your slides.
Okay. Good afternoon, all of you. Myself Pradeep Kumar. I am here to present our research work on comparative study. Open करिए sir. Comparative study of parasitic elements in snake headfish Channa punctatus, class Actinopterygii, order Perciform, from wetlands and river ecosystem at Lucknow, Uttar Pradesh. Now come to the introduction. Uttar Pradesh has a great variety, great areas of freshwater resources in terms of networks of rivers and canals, floods, plain wetlands, reservoirs, ponds, and tanks, which offer great potential for culture as well as capture fisheries. The state rank of Uttar Pradesh have third in land fish production after the Andhra Pradesh and West Bengal. It was contributing 7.57% in land fish production from India. Andhra Pradesh being the highest in land fish production, and it was contribute to 25.75% in the uh, total inland fish production of the country. The reason behind the less fish production in Uttar Pradesh compared to the top state may be attributed to the non-availability of quality fish seed, feeds, non-scientific fish farming practice, culture of only few selected species, and less subsidy for fish farming. Wetland aquaculture play a significant role in improving the economic status of the fish farmers and also crop getting better due to saturated field in India. The most abundant of aquaculture production comes from rural wetland aquaculture. Rural wetland aquaculture is the extensive or improved extensive system, low cost farming of aquatic organism by farming households or communities using technology appropriate to their resource base. Uh, Snakehead fish are widely consumed throughout Southeast Asia, China, and India because of their good taste of meat and high nutrient values, such as presence of prostaglandins, thromboxane, and omega-6 fatty acid. In the infection of helminth parasite in their body to change the quality of meat and uh, loss and economically losses in fish production and chain. So the parasite are one of the most serious limiting factors in aquaculture. Materials and methods. Fish sample were collected from two sites, wetlands and riverine of Lucknow, Uttar Pradesh. With also water sample were collected before and brought in the laboratory before dissecting the fish, taken the measurement of length and weight of the fish. After dissecting the fish and isolate the parasite from the different organs of the body, fish body for morphological studies. The elements of different groups, trematodes, cystod, nematodes, acanthocephalon, were isolated from the different organs of the host body. Further, trematode stand in Boris carmines and for morphological analysis, and cystod in Himalayan or Boris carmine. Nematode and acanthocephalon are stored in lactophenol for the morphological identification. Water parameters were also checked by temperature, pH, DO, hardness, alkalinity, chloride. DO is checked by Winkler methods, chloride checked by argentometric methods, and hardness is calculated the calcium and magnesium ion to or chloride is result and discussion. In overall investigation, the Hellman's infection in fish punctures so that females and males fish of wetlands were heavily infected than male and female fish of riverine with different groups of Hellman parasites. In this investigation, there are found 8 genera of Hellman parasite, in which 3 genera of trematodes, like Ginocarpus, Leopredium, Claniostoma, and nematodes. 3 genera of nematodes, Scarlet, Proclamulus, Eustranglides, and 1 genera of Senga and 1 Paliscentis nundae. The prevalence in first graph in this prevalence of Hellman parasite in wetlands. Female and male is high prevalence of the infection of parasite, and riverine is low infection rate than females. And second graph is number of parasite in male and female fishes of wetland and riverine. In this survey, the infection of the group of Acanthocephalus under Paliscentis, the high infection rate in females and males. Then Clinostomum. Then the second number Clinostomum are infected. And lengthwise parasitic prevalence in fish, more infection rate of 0.5 low length of the fishes, because but high infection rate in high length of the fish. But in this investigation, the number of fishes of less मतलब इसमें छोटी जो फिशेज हैं, उस छोटी फिशेज में इन्फेक्शन रेट जो है ज़्यादा है, मतलब विथ बड़ी फिशेज की अपेक्षा, क्योंकि इसमें जो बड़ी फिशेज हैं उनकी जो मात्रा है वो कम डिसेक्ट हुई है, मतलब कम जो है डिसेक्ट हुए हैं, लेकिन जो छोटी फिशेज हैं उनमें ज़्यादा हुआ है। तो इसी वजह से इस क्रॉप में जो है छोटी फिशेज की जो इन्फेक्शन रेट है उसमें ज़्यादा दिख रहा है, ठीक है। एंड बेट वाइज द parasitic prevalence in this higher weight of the fishes have more infected than low weight, and the water qualities of that both sides, wetland and riverine, have a little variations: temperature and pH, DO, hardness, chloride, alkalinity. Some parasites are isolated. This is the first figure, the Eustranglides, and second is the Eustranglide anterior portion, and second figure is the posterior region of Eustran, and third is the Euclaniostoma, and fourth is the Ginocarpus. Conclusion: in that overall survey, female fishes are heavily infected than male of wetland and riverine, and the water parameters have a little variations that is given in this table. So we can say that parasite as a bioindicators because the infection rate of the parasite in which the uh, water uh, to relation to the water parameters. Thank you. Thank you, sir.
Thank you, sir.
Thank you, Mr. Pradeep.
Thank you, sir.
It was a nice presentation. You have attended all the corners of the parasitology, from the morphology to the biochemistry. Okay?
Yes, sir.
But I may ask any query.
Yes.
Mr. Siddharth, may I ask any query?
Yes, sir, please.
Yeah, definitely. So, as you said, in terms of the biology, smaller the feces, little the infection, larger the feces, higher the infection. Am I right?
Yes, sir.
Could you explain the reason behind this entire scenario?
Because the infection rate of higher fish, big fishes, तो जो बड़ी फिश होती हैं सर, उसमें जो पैरासाइट के रहने के लिए जो अवेलेबिलिटी होगी, सारी चीजें जैसे नरिशमेंट उनको प्रोवाइड होगा, तो बड़ी फिशेस से ज्यादा उन्हें नरिशमेंट होगा, तो इस वजह से जो है पैरासाइट जो है उसमें रहने के लिए जो है सूटेबल ज्यादा होते हैं, तो इस वजह से नंबर्स उनकी जो है इन्फेक्शन रेट ज्यादा होती है विद कंपैरिजन स्मॉलर फिशेस.
Yeah, Mr. Pradeep.
Yes, sir.
The site or size of niche is important, or other factors may also collaborating to the rate of infection and infestation
Yes.
in these species.
Yes, sir. pH, waters, pH, CO2.
Yeah. What kind of the water quality parameter facilitate the more occurrence of parasites in the pisces? I mean, if you analyze the water quality,
pH.
then you will find pH.
pH. pH is more. pH and DO. pH and dissolve oxygen.
Dissolve oxygen. You are working on the river Gomti?
No, sir. River Gomti. Yeah, sir. Kukrel area जो है.
Kukrel area. Okay.
Yes.
Okay. Does any significant change in the pH in the different months?
Yes, sir. Change तो थोड़ा ज्यादा नहीं सर, बहुत little amount में change.
In the parameter means what are the minimum pH you have recorded and what are the maximum pH you have recorded?
Yes, sir.
What was the minimum pH?
Sir, minimum pH.
Yeah, minimum pH.
Sir, minimum pH 6.7 to 6.9 तक.
Okay. It means it is acidic in nature.
Yes, sir.
Okay. And have you did the work over the morphotaxometric analysis of Stringladis? Or based upon the microscopic analysis you have described it is a
only sir microscopic and molecular based.
Okay, what are the genes you have selected for the same?
Sir, 18 years and COX 1 and COE. ITS 1 and ITS 2. But sir, these parasites only 18 years and COE first.
Okay, thank you, Mr. Pradeep. Good luck.
Thank you, sir. Thank you, sir.
Thank you, sir. Anyone are ready? Dr. T. Lavanya or Anu Serawat?
Hello, sir. Myself Pungavi. Pungavi, sir. Yesterday you informed that
Yes. Please proceed. Your presentation, please.
Okay. Is my poster is available? Visible, sir? Sir? Is my poster is visible?
Not visible. Not visible.
Okay. Is it visible, sir, now? Is it visible?
Yes, now
sir?
visible.
Yeah, a warm welcome to this pleasant evening. Today I am going to myself Pungavi from Department of Entomology, Faculty of Agriculture, Tamil Nadu. And today I am going to throw some light on topic impact of pesticide residues on human health as well as environment and possible ways to overcome the threat for sustainable future. We all know that agriculture is a backbone of India ever since of origin of civilization. It has been the major task man to engage in some condition to improve his living conditions. The main source of human is food. The hunger was faced by before the Green Revolution about 1960. In order to increase the agricultural yield, farmers have tend to use up more number of agricultural chemicals, that is pesticides, herbicides, insecticides, that is rodenticides, etc., which is used worldwide to increase the agriculture productivity. But these pesticides, when it is applied on the crop, only 5% of this only reaching the target organism, that is pest as well as diseases as well as pathogen. About 95% of reaching the non-targeted organisms. FAO has given collected fruits and vegetable samples from markets. In that, they recorded the 85% of food sample containing at least some to permissible limit of residual limit. In that, 3 or more pesticides have crossing the maximum residual limit. That is for each crops, that is each goods, that is fruits and vegetables, even for any food grains, the maximum residual limit will pesticide residual limit will be given by Codex Alimentarius Commission. If it exceeds, it should not be consumed by the human. It causes ill effect in near future. Next, we are coming to see pesticide usage of global as well as India. Uh, the pesticide usage pattern worldwide among the pesticides, herbicides consumed by herbicides nothing but to control the unwanted plants, that is weeds, consumed about to control this 65% of herbicides are used, followed by insecticide, then followed by fungicides and bactericides. In world, US stands most consumption of herbicides, followed by Brazil. In case of India, Maharashtra stands first in pesticide usage for controlling the pest, because in India, insecticides is more usage, about 51%, followed by herbicide. Then coming to fungicides is used for disease control of disease. The overall pesticide usage among the crops is first it is cotton, followed by paddy. Next to that is maize. Till now, we have seen the pesticide scenario. Now we are going to see the impact of pesticides on human health. Usually, pesticides contain metals and heavy metals, that is arsenic, mercury, lead, cadmium, etc. These pesticides were applied to the plant by the farmers, mainly to increase the yield, at the same time to control the pest to kill the insects, which is causing damage to the productivity. But these pesticides, when it is applied to the plant, the pesticide residues get accumulated in the fruit. When it is harvested, it is taken to the market. Those residues were present in the fruit itself. When it is consumed by the human, it causes several ill effects. It may be acute or chronic. If it is acute, means sudden symptoms like vomiting, diarrhea, skin irritation. For example, most of the climatic fruits like mango, sapota are consumed by, or when it is harvested, then only they will go for ripening by using some ripening hormones. In such case, those chemicals, when it is applied during the process of ripening, when it is consumed by the human, it get entered into body and causes vomiting, diarrhea symptoms, that is acute symptoms. In case of when it is chronic disorder, when chemicals, these residue containing samples were consumed by the human, which leads to continuous accumulation of residues in the human body, and finally sometimes may lead to death. Here I listed some chronic disorder, that is cardiac failure, lung cancer, ovarian cancer, and musculoskeletal disease, and also renal dysfunction, and reducing thyroid hormone. These are the ill effects caused by the pesticide residues present in the food commodities what we are consuming day-to-day life. Here I have shown some case studies. Here, Helam and his co-authors in 2019, they collected samples. They collected both raw sample as well as processed sample. From that sample, they go for GCMS, that is GCMS analysis. In that, 88 pesticides residues were detected in their sample with 36%. Among the 8 pesticides, cypermethrin and followed by carbendazim, that is belong to organochlorine group, are detected more. In one fruit itself, several pesticide residues were detected. About 27% of multiple residues were detected in those samples. Among the 3 samples, tomato has having, that is raw tomato having several residue sample and compared to processed sample. Next, coming to investigations on analysis of cereals, food grains obtained from storage godowns, whereas cereals contains only samples obtained. In that, only 10% of residues were present. But in case of wheat, they're having more than 50% of residues in the grains. If this here they mentioned that the residual limit is higher, it is not applicable for human consumption. If they are consumed, they may having some chronic disorder in near future. And Neem and Sadish in 2016 also collected samples from the surrounding regions, where they obtained 34% of samples they collected contained pesticide residues. Apart from this, these pesticide residues not only affect the human health, and also it affects the environment, which leads to bioaccumulation, biomagnification. Bioaccumulation is nothing but overuse of pesticides, which at higher concentration get accumulated in the soil from microorganism to macroorganism, which in turn leads to biomagnification. Increased concentration of these accumulated pesticides in soil as well as any microorganism, but to some extent they affect the food chain, the overall biodiversity get affected. Here I finally come to conclusion. There are possible ways to overcome to threat. Go for organic foods, or buying of organic goods, organic fruits and vegetables, or go for gardening gardening, where you can use your own vegetable waste for their increase the yield, instead of using the chemicals to control the pest, or use some botanicals, some pest or tree neem leaf extract can be used for spraying instead of insecticides, chemical insecticides, so that we can control the pest. And we can also obtain organic goods from your garden itself. And the washing of the food products is very essential, because for example, we are consuming apple, they are having wax coating. They may also affect our human body once they get accumulated day to day life. And also same like mango, sapota having climatic fruits, grapes. In grapes, they are affected by several pests and diseases. They go for using of several insecticides. They get accumulated in the fruit and get entered and once they are taken to market, we are consuming without washing, to surely they will the chemical get accumulated in our body. To some extent, to reduce these pesticides, we can go for washing. And also processing of food is essential. Cooking will reduces that is boiling of any food grains will reduces about 35 to 60% of organophosphorus chemicals. In case of 20 to 25% of organochemicals, that was I mentioned, cypermethrin, carbendazim can be reduced. If the grains having these chemicals can be reduced if you go for boiling. In case of farmers' point of view, go use of rational use of pesticides. That is recommended dose of pesticides can be used for control, so that we can reduce the residues in the soil. And the use of natural insecticides, that is neem leaf extract, ginger leaf extract, etc. and also implementation of laws, that is pesticide law given by Codex Alimentarius Commission. These commissions are given residual limit for each commodities, so retailers can test their products so that we can avoid the residues in their commodities. Finally, I conclude that the processed food and the washing of food products and go for organic can be healthy, uh, can be done, so that to some extent we can reduce the residues in our day-to-day life food, which leads to healthy life for sustainable future. Thank you, sir.
Thank you. Thank you, ma'am.
Thank you, sir.
Next participant, Syed Khalida Ijhar, Integral University, Lucknow.
Hello. Good afternoon.
Yes.
Sir.
Good afternoon, ma'am. After that, next participant is Anu Serawat. Please be ready. Miss Syed Khalida Ijhar. Miss Syed Khalida Ijhar.
Yes, sir. It is visible.
No, not visible.
There is a network issue.
Next participant, Anu Serawat. Are you ready? Miss Anu Serawat, please proceed if ready. Yes, Miss Anu Serawat, please proceed. After that, Syed Khalida Ijhar will present. Miss Anu Serawat. Next participant is Sonia. Next participant Miss Sonia, Department of Chemistry, MMDU Mulana. Next participant Miss Sonia. Next participant Dr. Parmeet Kaur. Next participant Dr. Parmeet Kaur, Chandigarh University. Next participant Nending Muni. Next participant Nending Muni, Department of Zoology, Rajiv Gandhi University, Arunachal Pradesh.
Yes, yes.
Are you ready?
Yes, yes.
Please,
Am I
please
audible?
proceed. Yes, yes, you are audible. Please proceed.
Okay.
Nung baca kepala nung. Oh. Aku sini baca saya mana?
Nending Muni,
Yes, sir.
please share your slide.
Sir, I'm unable to share. Just a minute. Share tuh ada nggak ya? Share. Open your cell ke ada. Share kan? Oh no. Is it visible?
Yes, visible.
No. Is it fine now, sir?
Yes, yes, fine.
Okay. My name is Nending Muni. I'm a PhD scholar from Department of Zoology, Rajiv Gandhi University. And the topic of my presentation is volatile profiling of host plant and EAG responses of edible shepherd beetle, that is Anomala species. So this Anomala, it is a Phytopegus beetle, which is commonly known as a leaf chafer, or it comes within the white grub fest, and it is a characteristic metallic coppery green color. This species is cosmopolitan, and it is distributed throughout the India. And in the northeast region, it is reported in Arunachal as pest of various crops like kiwi and chestnut plants. And these groups are economically important, with leaf defoliating adults and their larvae feeding on various ground crops. And in addition to this being a pest, it is also reported as an edible insect, and it is consumed by the ethnic people of Northeast India. So in this perspective, this beetle is quite important as a pest as well as for edible purposes. This one is the picture of Anomala dimidiata, male, dorsal view, and a lateral view. So these are the Phytopegus chaper beetle, and many sex pheromones and allelochemicals with conformation has been identified for this species, and it is already promoted for biomonitoring and also as pest management in agroecosystem. So as such, many leaf-derived allelochemicals have been identified for several animal species as well. So the purpose of our study is to examine the presence of volatile cues in the host plant of this beetle, which can be further applied in agriculture or biomonitoring perspective. So this one is the graphical extract abstract. Out here we have taken the host plant. This one is Castinopsis indica leaves, and we have done the solvent extraction and the essential oil extraction, and also this is the headspace SPME analysis in GCMS. All of them was analysis in GCMS, and the compounds was identified based on this library. And on the other hand, the solvent extract and the essential oil extract, they were gone through EAG assay, and we tried to figure out the responses of the beetles antenna. So materials and method, this insect were collected from the host plant from the old geographical locality. It is located in Lower Subansiri district, Arunachal Pradesh. And for identification purpose, we have referred specimen from APRC GDSI. This is Arunachal Pradesh Regional Center, which was examined by Chandra and Gupta in 2012, and the pin specimens were also deposited at the same. For the solvent extract, we have used the method of Jade et al. With few modifications. We have taken 250 grams of set dried leaves, percolated with methanol for 3 days, and we have fractionated it from non-polar to polar. And the yield, it was taken out and concentrated in Effender concentrator plus and kept in the fridge for further analysis in GCMS and EAG. For essential oil extract, we have used hydrodistillation process in Clevenger apparatus. It was maintained in a normal boiling range of water and atmospheric pressure, and the extraction process, it was adjusted to around 6 hours with the heating power of 350 watt and 1:2 solid to gram solvent ratio. And solvent, we have just used the distilled water, and the oil, it was dried using this anhydrous sodium sulfate, and the calculation was done using this formula y equal to b into 100 whole divided by w. And for headspace SPMA extraction of leaf, we have just taken the fresh leaf and put it in a vial and seal it with PTFE septum, and the SPMA fiber, it was exposed for 30 minutes for the equilibration of the volatile component, and it was directly injected into the GCMS. The GCMS analysis of this extract and essential oil, it was diluted into 1:100 ratio, and 0.5 microliter was injected directly into the GCMS, and the analysis was carried out in thermoscientific gas chromatography, and it was equipped with TSQ 8000 mass spectrophotometry with FID detectors. And this volatiles was identified by comparing the mass spectral data of the NIST, National Institute of Standard Technology. Sorry. The EAG recording, EAG assay was done in a 2-channel signal acquisition interface IDEC2 system, in which is the product of Syntex Netherlands. And the EAG preparation was prepared as described by the Olson and Hanson 2013. The antenna was removed at the base and it was fixed at the electrode probes, and the extraction stimulus was applied using a stimulus air controller type CS55 Centix Netherlands, and the constant airflow was also maintained at 4 to 5 minutes per 4 to 5 meter per second with complementary air supply at 4 meter per second connected to the stimulus applicator. So IDEC2 Syntec signal acquisition system was used for signal acquisition, and EAG Pro Syntax, it was used for processing data. We have done some dosage response trial, and from there we have found out that 10 microliter, which is 80% of the extract, it gave a good response. So it was selected as the optimum dosage for all the EAG recordings. And the hexane alone treated filters type, we use this as a control, and we have taken one reference to that one is Z3 hexane 1-ol. It is a non-attractant of Anomala species. This responses, it was measured and presented in graphical form, and we have done also its percent response. It was given by Jyoti et al. Like in this process, we take the reference percent as 100, and with compared to that, we convert all other recordings and compare the percentage with S of the reference. So the result, this qualitative GCMS analysis, it has shown that the solvent extract, which is the hexane fraction of the leaf of C. indica, revealed that there is 76 VOCs, volatile organic compounds, with representing 93.75% of the total content. And the same, the essential oil extract also gave 78 compounds representing 96.85% of the total oil. It is given in table 1 and 2, and the profile by headspace SPME GCMS from GCMS also it's provided in table number 3. This one is the volatile compositions from the solvent extract of C. indica, chromatogram, and table provided some compounds. And this one is for the essential oil composition, same C. indica, chromatograms, and the tables. And this one is the volatile composition from HSSPME leaves of C. indica. Now the EAG responses, we can see that this one is the solvent extract, hexane fraction, and the essential oil extract. With compared to the reference, we have considered it as 100%, and the essential oil it shows above 60%, and the solvent extract it shows above 50%. This one is the EAG recordings. We have given the 2 replicates. This one is for the reference hexanal 3 Z3 hexane 1-ol 3 replicates, and this one extract is for solvent extract 3 replicates, and essential oils, and the controls we have just given 1 for each of them. So data analysis, it showed that ANOVA, we had done one-way ANOVA, and it showed that there is a significant difference in response by in the solvent extract and essential oil extract as compared to the control. This diagram is some of the allele chemicals of Anomala species which is present in C. indica leaf extract. Hexane 3 hexane 1-ol, it was present in essential oil, and trans-tunnel, it was also present in essential oil as well as in SPME headspace, and it is present in SPME of lips. So discussion, the result of this present study showed that the antenna of the Anomala beetle saw good responses to the solvent extract and essential oil extract of this host plants, C. indica, which implies that there must be some volatile organic compounds or essential oil presence which elicits this response. The literature review showed that trans-2-nonenal, which is present in essential oil extract, is a non-attractant to Anomala sconfelti, and also Anomala elevophilosa and Anomala dubia. and this Z3-hexane-1-ol, which is present in essential oil as well as in SPME of leaf, is also listed as allyl chemicals with attractant effect for Anomala corpulenta and Anomala exulata, as well as for Anomala actis scutata. But no any attractant were present in the solvent extract of the GCMS. This anethol, it was present only in the SPME of fresh leaf, and it is also a non-cardamom of A. octis scostata. So the conclusion is this research effort, it was mainly driven by various reasons that this chapel betel is edible, as well as it is found in Arunachal Pradesh, and its host attractant can be simultaneously explored and applied in the biomonitoring of these beetles, spatial and temporal distribution with using this host plant-derived attractant. And in case of invasive species in some regions, it can be used in mass trapping and pest management strategies for many crops. The BOC that are present in the extract may also contain some chemical attractant to these predators, which can apply in population control of this type of beetles with natural enemies via plant-induced signaling in response to herbivore damage. There is some limitation in this research because the typical function of this EAG, it's kind of like qualitative in nature. So its result can just be seen as an opening for possible subsequent essay in GCEAD, which is more efficient.
आप डाउनलोड करा था रोहित केपी क्लासेस का, तो मैं जान सकती हूँ आप कोई कोर्स देख रहे थे या फिर किसी और पर्पज से आपने आप डाउनलोड करा था?
So further studies on the different exploration of identification of allyl chemicals and their field application can
कोर्स देख रहे थे या फिर आपको किसी ने बताया डाउनलोड
the
करने के लिए,
reference.
आप मुझे बताओ.
Thank you.
ओके, वो जो 51 डेज के जो यूट्यूब वीडियोस पड़े वो है ना? ओके, तो आप वीडियोस देख रहे हो अभी या फिर आपको कोई कोर्स मतलब क्लासेस भी करने थे?
Hello?
आपको कुछ करना है, लेना है हमारे से क्लास?
No, ini ko na.
Hello.
Hello?
Hello. Miss Nending Muni.
Yes, sir.
Your presentation is complete.
Yes.
Okay. Then next participant is Miss Sonia, Department of Chemistry.
Yes, sir. Good evening, sir.
Yes, please proceed.
Okay. Can I share my screen?
Yes, please.
Okay. Am I audible, sir?
Yes, very much audible. Please share your slides.
Okay. Good evening, one and all present here. I am Sonia, research scholar at Mehersi Markandeshwar deemed to be University, Mulana, Ambala. I am here to present a report on application of nanomaterials to improve phytoremediation for the 6th International Conference on Recent Trends in Life Sciences and Biotechnology 2022. So yeah, first of all, we will start from the introduction.
Your slide is not visible.
It is not visible. Is it visible now, sir?
No, no. Yes, visible.
Okay, sir. So first of all, we will start from the phytoremediation. What is
Yeah, please.
phytoremediation?
Open your title slide first, madam.
Okay. Yeah. Good evening, one and all. I'm Sonia, here to present on applications of nanomaterials to improve phytoremediation for the 6th International Conference on Recent Trends in Life Sciences and Biotechnology 2022. I'm doing research at Maharishi Markandeshwar University under the supervision of Dr. Nadeem Sharma in Department of Chemistry. Yes. First of all, we will start from the introduction. This is phytoremediation. So phytoremediation includes the efficient use of plants for the removal, detoxification, or immobilization of environmental contaminants in a growth matrix such as soil, water, or sediments through biological, chemical, and physical plant activities of the plants. And various methods have been developed in this regard, but nanomaterial based technology shows the most promising behavior as we can observe by the literature survey we have mentioned below. So here are the sub processes which are included in the phytoremediation, like phytovolatilization and phytodegradation, phytoextraction, phytostabilization, phytostimulation, and rhizofiltration, as we can see in this plant picture. So if I will explain in brief what is phytoremediation. So it is an innovative use of green plants to clean up our environment. So the term comes from Greek word in which phyto stands for the plants that can detoxify or remediate soil or water contaminated with hazardous metals or excess minerals. Basically, it stabilizes or it decontaminates the polluted areas using plants. So here are various applications of nanomaterials in the process phytoremediation. So first of all, it can directly remove the pollutants from the soil, water, or other bodies. So in this context, carbon nanotubes play a significant role due to their exceptional adsorption capacity. They can stabilize organic pollutants via hydrophobic or electrostatic interaction, whereas inorganic pollutants require complexation or physical adsorption. In the next application part, as plants growth promoters. So nanoparticles can play a very big role in the plant's growth to promote promote the growth of plants. So as studied by Praveen et al., that the role of zinc oxide nanoparticles as plant growth regulator by suppressing cadmium and lead phytotoxicity in white popinac tree. So they have studied our white popinac tree. In the next application, the nanomaterials can enhance the phytoavailability of the pollutants. So as have been studied that C60 fullerene nanoparticles for eastern cottonwood phytoremediation to improve trichloroethylene from soil, which increase the uptake of pollutant by 30 to 65%, which is mainly in the roots. Nanotechnology can also play a very good role in heavy metal removals. So it can help in removal of heavy and hazardous metals ions as described here. Nanohydroxyapatite in phytoremediation of lead. So nanohydroxyapatite can be used in the removal of lead. Phytoremediation efficacy of ryegrass was subjected to nanohydroxyapatite effect. Results were analyzed after few months, which can be which are given by Liang et al. and also iron oxide nanoparticles for removal of mercury. In an analysis, 3 types of nanoscale zero-valent iron, nanomagnetite, and iron sulfide nanoparticles were analyzed to treat a soil contaminated with mercury ions. The nano zero-valent iron particles were boomer, which immobilize 98% of the mercury ions, while the other two can cure only up to 85 and 80%. So here, the nano zero-valent iron play a very important role in the immobilization of mercury ions. Although all the 3 are mastered, but the choice depends on conditions maintained around. Zinc oxide nanoparticles also play a very good role in removal of highly toxic metals like cadmium and lead, which can be reduced by zinc oxide by the regulation of some physiological activity of the plants. Silicon nanoparticles can be used for the removal of chromium metal ions. Chromium phytotoxicity on pea plants were studied, and in the form of remedy, silicon nanoparticles were applied, which minimize the stress level created by chromium ions. Titanium oxide nanoparticles. In the first step, nanoparticles occupy the chloroplast region and increase the light reactions going on in the plants. The performance is also increased by titanium oxide nanoparticles. Similarly, aluminium, zinc oxide, and zinc nanoparticles can affect on the root system and germination of some plant species like corn, cucumber, lettuce, radish, rape, and ryegrass. Also, copper oxide nanoparticles can change the photochemistry of Chlamydomonas reinhardtii green by altering its various photosynthetic reactions. Salicylic acid nanoparticles can decrease the arsenic concentration of up to 80% within a period of 5 weeks only. Some other nanoparticles are also like iron oxide, iron containing bimetallic species, natural mineral, phosphate based substances, which are helpful for the removal of impurities. Hydrogel supported hydrous iron oxide nanoparticle was studied for the removal of ions like lead, copper, cadmium, and nickel from polluted water. Their order was like this, which is comparatively higher than studied by Beiringer. Now, this study is also based upon the choice of plants. So the success of phytoremediation is decided by uptake percentage by the plants, its growth rate, and plant biomass. Graphene, carbon nanotubes, silver nanoparticles, gold nanoparticles, zinc oxide nanoparticles, and nano zero-valent iron particles and upconversion nanoparticles are used, but still a limited quantity can be absorbed by a particular species. So in this slide here are some advantages and limitations of using nanoparticles in the phytoremediation process. So here, as we can see, the great diversity of tropical plant yet to be explored in this study. It can be applied in large areas. It depends on solar energy, erosion reduction, waste generation, and emissions are minimal, low cost, and ecologically accepted. Some plant species are well established and adapt to heavily polluted soil, beneficial soil microorganism associated with plant roots, and hence heavy metal photoremediation. But here are some limitations also, like physiochemical soil properties. Soil inputs of nanoparticles and nanofertilizers is very less, and tolerance to heavy metal under certain conditions is also very low. Morphology and depth roots, these are the conditions which can decrease the use of nanoparticles in photoremediation. So in conclusion, we can say that nanomaterials are a booming idea, but in photoremediation, where both fields work together, photoremediation with the help of nanoparticles is an economical and attractive technique growing in the recent era. This technology not only helps in the removal of heavy metals pollution, but also help in the plant growth, thus increasing the biomass. Nanomaterials generally enhance the phytoavailability of the heavy metal and also encapsulate that metal in the plant system. So this is all about the nanomaterials used in phytoremediation. Thank you so much for the patient listening.
Thank you
Hello,
so much.
am I?
Yes, yes.
Okay, thank you, sir. Thank you.
The next participant is Disa from Saloni University.
Hello.
Yes, yes.
Am I audible, sir?
Yes, audible.
Can I share my screen, sir?
Yes, yes.
Is it visible, sir?
No, it's not visible. Yes, it's visible.
Okay, thank you, sir. Good afternoon, everyone. I'm here to present my presentation on
No,
topic
it just
entitled.
vanished. Please again.
Hello.
Yeah, it's vanished. Please again.
Okay, sir. Okay, sir. Now it's visible, sir.
Yes, visible very much.
Okay, sir. Should I start, sir?
Yes, please.
Good afternoon, everyone. I'm here to present my presentation on topic entitled Phytochemical and Antioxidant Assessment of Thalictrum folliculosum and Cordia dichotoma, traditionally used against jaundice. Myself, Disha, and I am a PhD research scholar in Shulini University under the guidance of Dr. Rachna Verma. First of all, I will start with introduction. The use of herbal medicine is an age-old tradition and are considered to be of great importance among different rural and indigenous community in many developing countries. According to World Health Organization, as many of 80% of the world population and in India 60% of the people of rural area depend on herbal medicine as their primary healthcare system because of better culture acceptability, better compatibility with human body and their lesser side effects or very low side effects. Traditional knowledge of medicinal plants and their use by indigenous healers in the present day are not only useful for conservation of cultural tradition and biodiversity, but also for community healthcare and drug development in local people. The scientific study of this plant-human relationship is termed as ethnobotany, which deals with various aspects, in which one of the most popular and common aspect is the study and use of herbal medicine as called as ethnomedicines. Now, aspects of jaundice. Jaundice is not only a disease but a symptom of liver indicating the liver malfunctioning or dysfunctioning. It is also known as eye-steris, which means yellowish pigmentation of the skin, mucous membrane, and sclera due to the increased level of bilirubin in the blood. Basically, bilirubin is a natural product arising from the normal breakdown of red blood cells, also called as erythrocytes, and is excreted in the bile duct through the action of liver. Based on the pathophysiology, jaundice is of three types, that is prehepatic, hepatic, and posthepatic jaundice. Generally, prehepatic jaundice is caused due to excessive breakdown of red blood cells, whereas the hepatic jaundice is caused due to the any obstruction in the liver or any malfunction of the liver, whereas the posthepatic jaundice is caused due to any kind of obstruction and obstacles present in bile duct and secreted by bile duct. Now, effectiveness of herbal medicines in treatment of jaundice, basically correlation of antioxidant and its impact on treating jaundice. The application of the medicinal plant in treatment of jaundice is conspicuous from the number of case series which express faster reduction of jaundice than Western medicine treatment. Furthermore, there are very less medicines or also no in allopathy and homeopathy, no treatment is available against jaundice. Only some steroids and immunosuppressants are used for the treatment of jaundice, which, that is very costly, and hence herbal medicines has gained popularity due to their side effects and costly effects. Various active phytoconstituents are responsible for the therapeutic effect on jaundice belonging from different chemical categories. such as flavonoids, phenols, alkaloids, terpenoids, compounds that contribute and attribute to the antioxidant effect of plant that act as a defense sheet and against various diseases. The effectiveness of phytochemicals in the treatment of jaundice may lie in their antioxidant effects because
Excuse me, Miss
antioxidant.
Lisa.
Yes, sir.
Please change your slide.
Now, okay, sir.
No. Yes, yes, okay.
So the
Yes, please
effectiveness.
proceed.
Okay, so thank you. Antioxidant can delay, inhibit, and prevent the oxidation of oxidizable materials by scavenging free radical and free radical species and diminishing oxidative damage by and prevent damage to important cellular components such as DNA, proteins, and lipids. Based on the antioxidant effects on its high efficiency, there is increase towards the use of herbal and natural products in the treatment and prevention of many diseases, including jaundice. So to achieve our study, the following objectives has been selected. First of all, the survey and documentation of ethnomedicinal plants used for jaundice treatment in Solan district, Himachal Pradesh. Second one, on study on their phytochemical properties on two important selected plants. Third one is the analysis of antioxidant activity or biological activity of these selected medicinal plant extracts. Material methodology include, first of all, the study area. The survey has been conducted in 7 different tehsils of Solan district, including as shown, as indicated by the red stars in map, that is including Solan, Kandaghat, Kasauli, Nalagarh, Arki, Baddi, and Ramshahar. Thereafter, the ethnobotanical survey of plants used by rural people of district Solan for treatment of jaundice and the plan of work and the methodology to be adopted is presented below, including field survey, collection of samples, preparation of herbarium, and identification of specimens. Thereafter, the selected plants has been taken to the laboratory for further biological and phytochemical analysis. For that purpose, rotary orbital shaker method has been used for the extraction, and 3 different solvents including methanol, chloroform, aqueous based upon their polarity, that is polar, non-polar, and aqueous solvent is used on the basis of traditional use, that is according to the survey. Thereafter, the qualitative and quantitative analysis of the selected plant extract has been done using different parameters and methodology. And thereafter, the identification of dominant compounds using method of UHPLC analysis has been used. And thereafter, the third objective, that is analysis of biological activity, dominantly antioxidant activity using 3 different assays, DPPH, ABTS, and PrEP assay has been done. Now results. So as I told you, survey were conducted in 7 different tehsils of Solan district of Himachal Pradesh. It has been observed from the study that there are total 31 traditional healers using using different ethnomedicinal plants for treating jaundice in their own different way. Among these 31 healers, 29 are males and 2 are females belonging to different age groups, include from 35 year to the 88 year. Ethnobotanical survey on the present study revealed that 26 different ethnomedicinal plants are utilized by these indie healers and local healers in Solan district. So these are the picture of those 26 ethnomedicinal plants used by the local healers for treating jaundice in their own traditional way in district Solan, Himachal Pradesh. Thereafter, the collection of these plant samples, the herbarium has been made and plants are identified from the DSI Dehradun. Among 26 different plants, only 2 plants have been selected for further laboratory studies, including Thalictrum folulosum and Cordia dichotoma. Thalictrum folulosum, commonly called as Chambra and Jhirmir in Solan district of Himachal Pradesh, belonging to family Ranunculacea. Based on 3 different criteria, this plant has been preferred for further study. First of all, on the basis of survey data, it has been observed that roots and leaves are highly utilized and effective according to traditional healer against jaundice of this plant. And second one is the according to Ayurvedic background. So it has been observed and it has been shown that this plant is considered as very good substitute of plant kutki, especially the roots of this plant. Kutki, botanically called as Genetiana kuro, that is considered very effective and widely used to treat epidemic jaundice, but now in endangered condition. That is why the plant is taken as alternative for the Genetiana against treatment of jaundice. And third criteria was according to literature. It has been observed that there is very less work is available on this plant and kind of unexplored plant. Second one is Cordia dichotoma, commonly called as Lashuda and Lasura in Solan district of Himachal Pradesh. Also, there is again three criteria selection of these plants. First of all, the survey data. Survey data show that the leaves of these plants is highly effective and show great potential against treatment of jaundice. Whereas on the basis of Ayurvedic background, it has been shown that it is known to have good gastroprotective potential or having specific pitta dosha. Pitta in Ayurveda, pitta is called for the chronic or any liver diseases karma in Ayurvedic based literature. And third one is according to already published and present literature. Very less work is found on especially on the antioxidant and in vitro activity of Cordia dichotoma. Although very less papers are available that show curative potency of these plant against jaundice. So after selection of the plant, and they have been taken to the laboratory, and using different solvents, different extract has been made. First of all, qualitative phytochemical analysis of selected plant has been done. First for methanolic, and these tables showing uh, the qualitative phytochemical analysis of selected plant extract using different methodology and parameters, that is different presence of to showing presence of different phytoconstituents present in selected plant extract. And this is the qualitative phytochemical analysis of selected plant extract of aqueous extract. Thereafter, the plant has been taken. After checking the presence of the different phytoconstituents, they have been taken for quantitative phytochemical analysis. Uh, so figure A depicting the total flavonoid content, in which highest flavonoid content has been shown by the CDLM, that is Cordia dichotoma leaves methanolic extract, whereas the lowest flavonoid content has been shown by the TFLA, as shown in the figure, that is Thalictrum folulosum leaves aqueous extract. Same in the case of figure B, that is showing the total phenolic content. Figure C depicting the total tannin content, where again highest content has been shown by the methanolic extract and lowest tannin content shown by the Thalictrum folulosum leaves aqueous extract. Figure D depicting the total saponin content. There is the figure E depicting the total alkaloid content, and figure F depicting the total carbohydrate content. Last one is the figure D, that is depicting the total protein content. So overall, it is concluded that the methanolic extract showing the highest amount of different phytoconstituents, especially the flavonoid and flavonoids and phenols, that is predominantly responsible for the antioxidant activity. Thereafter, the qualitative and quantitative phytochemical analysis, the plants has been taken for their identification and characterization for checking the dominant phytoconstituents. So UHPLC analysis has been done for identification of dominant phytoconstituent. It has been shown that the CDLM, that is Cordia dichotoma leaves methanolic extract, show the highest amount of phytoconstituent, that is routine in flavonoids, whereas in TFRM, caffeic acid show the highest amount of phenolic compound. So these are the UHPLC chromatogram of figure A depicting the UHPLC chromatogram of TFRM, that is Thalictrum folulosum roots, whereas figure 2 depicting the UHPLC chromatogram of CDLM, that is Cordia dichotoma leaves methanolic extract. Figure 3 depicting the UHPLC chromatograph of again TFRM, whereas figure 4 depicting the UHPLC chromatograph of CDLM. Thereafter, the results of assessment of antioxidant activity. So different assay has been used, that is DPPH, ABTS, and FRAP assay. So first of all, DPPH assay, it has been shown that the highest antioxidant potential of DPPH assay has been shown by the TFRM, that is Thalictrum folulosum roots methanolic extract, with IS50 value of 5.95 microgram per ml. It is known that the IS50 value is inversely proportional to the drug potency. Lower the IS50 value, higher will be the potency of the drug and the extract. So our extract has been compared with the standard, that is taken as ascorbic acid. Second assay was the ABTS assay. Same in case of ABTS assay, IC50 has been calculated and compared with the standard, that is gallic acid, and it has been shown that the highest antioxidant potential was shown by the CDCl, that is Cordia dichotoma leaves chloroform extract. Thereafter, the third assay that has been performed to check the antioxidant activity and potential of selected plant, that is FRAP assay, that is ferric reducing antioxidant power assay. So it has been shown that the highest antioxidant potential extract was the CDLM, that is Cordia dichotoma leaves again methanolic extract, and it is compared with the standard, that is ferrous sulfate. Discussion. So despite effective traditional utilization and medicinal value of selected plants, that is Thalictrum and Cordia, for treatment of various liver illnesses, these plants are still unexplored for its scientific and chemical profiles. As a response, the current study is an attempt to examine the antioxidant potential of the prospective plants based on local healers' beliefs and folklore. Throughout the field research and interaction with the locals, it was observed that the villagers make the use of traditional medicines with complete faith and confidence, since they had learned from their elder generations or their own personal experience. Overall, the study revealed that the methanolic extract showed the maximum amount of phytoconstituent as compared to the chloroform and aqueous extract. Also, the discussion demonstrated that the Thalictrum folulosum roots and Cordia dichotoma leaves contain the substantial and the significant potent bioactive compounds and potential antioxidant activity. Now, the conclusion and future recommendations. The methanolic extract of Thalictrum folulosum roots and Cordia dichotoma leaves possess substantial antioxidant properties that make them potentially useful for pharmaceuticals use on an industrial scale and innovative treatment for a variety of liver diseases, especially jaundice. With this point of view, following recommendations are suggested from the current research. First of all, there is an emerging interest in the use of medicinal plants that have been used traditionally as Thalictrum and Cordia for treating jaundice, but more epidemiological studies and clinical trials, including preclinical studies, that is in vivo and in vitro cell line studies, base studies, are required for the practical implementation of the plant pharmacotherapy of jaundice, especially in Himachal Pradesh. Additionally, a conservation strategy must be developed to preserve significant medicinal plants that people in rural areas use as a part of their folklore use, and to raise awareness in order to motivate them to cultivate more and more of these important and more medicinal plants on a larger scale in order to promote their economic and ecological development. Lastly, I would like to acknowledge my supervisor and my guide, Dr. Rachna Verma. Thank you.
Thank you so much, ma'am. But your slide is just freezed all over the presentation.
Should I present it again, sir?
No, it's okay.
Okay,
and the last participant again, Miss Anu Serawat.
Hello, sir.
Yes.
Yes, sir. I am audible, sir.
Yes, you are audible.
Thank you so much, sir. So due to some network issue, I am facing problems, so I am wasting your precious time. So sorry, sir.
It's okay.
Okay, sir. Good evening, everyone. Respected sir
Please
and ma'am.
conclude within the 10 minutes, please.
Now my poster is visible, everyone.
Yes, visible.
Okay. Good evening, sir. Respected sir and ma'am. I am Anu Sharawat, PhD scholar from Microbial Biotech. I am doing research work from Central Soil Salinity Research Institute, Karnal. My supervisor, Dr. Mukesh Yadav, Assistant Professor of MMDU, Maharishi Markandeshwar University. My co-supervisor, Dr. A.K. Rai, Principal Scientist of ICR CSSRI, Karnal. My topic is arbuscular mycorrhizal fungi potential tools to ameliorate the plant growth in salt stress soil. Now you know everyone, presence of salt in soil is one of the biggest problems that were considered among the biotic stress factor which decrease the unusability of agriculture lands. And salinization of soil is a serious problem and it increasing steadily in many parts of the world, in particular arid or semi-arid regions. Saline soils occupy the 7% of the earth land surface and increase salinization of areas and will result into a 50% land loss by middle of the 21st of the century. At present, out of 1.5 billion hectares of the cultivated land around the world, about 77 million hectares, 5% is affected by the excess salt content. High levels of salinizations greater than 4 in soils is mainly due to the soluble salts present in the soil and in irrigation water used in agriculture. Excessive salts in soil affect the established growth and development of the plant, leading to huge losses productivity. Arbuscular mycorrhizal fungi colonize the plant root system and modulate the plant growth in various ways and also play a significant role in elevations of soil stress as well as plant growth and productivity. Arbuscular mycorrhizal fungi are the obligate biotopes that establish the mutualistic associations with roots of most terrestrial plants, including many crops. These roots colonizations by AMF fungi improve the uptake of mineralizations and the nutrients in the host plants, mainly phosphorus, nitrogen, and exchange for photosynthetically mixed crops. Now, mechanism of this arbuscular mycorrhizal fungi, mainly due to salt stress, is all of this abiotic stress, including salinity, can cause 50% production losses. And these AMF fungi occur majority in natural habitat, and they provide range of important ecological services, in particularly by improving the plant nutrition, stress resistance, stress tolerance, and soil structure and fertility. These stress that affect the plant growth and development, plants are sessile and have development sustainable mechanisms to these high salt environments. Salt stress basically to growth inhibitions and cell death during prolonged exposure. Along this adverse effect of salinity on plants, this toxicity of excessive sodium, calcium, ions towards the cells, and salinity cause physiological droughts, damage to the structure of the enzymes and macromolecules, disruptions of the photosynthesis, respirations, and protein synthesis. All these nutrition imbalance caused by the nutrition uptake and transfer to the shoots, leading to an iron deficiency. We all these know most common types of stress in plants. Basically, they are hazardous to the stress: temperature stress, salinity stress, water stress, mechanical stress, stress due to absence or excess of nutrition, stress produced by infections from pathogens, pests, and viruses, stress due to lack or excess of lights. All these salinity stress, ionic stress, and osmotic stress. Now in this figure, we have in second figure stress tolerance in rice and wheat, physiological and molecular mechanisms. In this figure, osmotic effect and water deficiency effects. These are the reduce the plant's ability to take up water, and this leads to slower the growth of the plant. And salt specific effect are those effects also known as iron excess effect. Salt enter the transpiration streams and eventually injure cells in the transpiration leaves, further reducing the growth of the plants. They have biochemical changes, morphological changes, physiological changes, and biomass grain yield. In biochemical changes, we consider oxidative stress and lower the zinc and potassium uptake. In physiological changes, in ionic toxicity, inhibitions of photosynthesis lower the osmotic potentials. In morphological changes, ionic toxicity again, and stunting to the plant growth and poor root growth productions. Biomass yield grain of the plants is low, less grain yields also. Again, all in these accumulations of the salt stress, they compete the plant growth, salt stress in plant growth and their development. For examples, in affect the plant growth can measure by the decrease in the plant growth rate and decrease also the biomass accumulation. Leaf elongations due to the cereal's responses of the hyperosmotic stress within the seconds, one of the most sensitive plants responds to the stress that all disturb the plant growth and yield formation. Thank you for listening.
Thank you
Hello.
so much, ma'am.
Thank you, sir. Thank you for listening.
Thank you. This is the last presentation. Now no presentation are there. And I would like to invite you, Dr. Sushil Kumar Upadhyay, sir, to conclude the meeting and today's seminar.
Thank you, Mr. Siddharth.
Thank you, sir.
Yeah, it was very exhaustive day for the today, even though the opening is quite not easy task. But when a good opening, then it means half the task completed. So we have right now completed our opening task and the day complete task. You have planned a entire session very well. And there were lots of the presentations by the eminent scholars, as well as the delegates from the different disciplines from the different universities across the different states of the India. And I suppose few of them were also from foreign countries as well. How much the entire presentation was today?
Sir, approx 23-24.
Yeah, it is more than 20 presentation was delivered by the young scholars, and it was keenly observed by the recognized jury members throughout the session. And it has been marked accordingly based upon the content of presentation, mode of presentation, way of interaction, query resolved, query answered, as well as the entire view and affirmative social impacts of the research that was presented by different scholars. So based upon these few criteria, the reputed jury members has been well documented the entire evaluation. And in future days, 2 days, the similar kinds of activity will be going on. And in the last day, we will try to evaluate and compare entire presentation to give the medals to the suitable candidates, which will get the maximum. So maximum 6 candidates will be awarded by the gold medals, silver medals, and bronze medals in different categories. And the delegates that were presented the sessions today, I'm very extremely thankful to them also. From apart from the India with different states, they joined this entire session, and I'm extremely thankful to our guests during the inaugural session. The administrative authorities giving me permission to host this kind of event in the coming 2 days also. And I'm very much thankful to our HOD for the HOD sir for the motivational encouragement and all kinds of support during this. So he's working as a backbone of entire session. Without him, it was not possible to organize this kind of huge event, in which about more than 21 countries and 27 states of the India has been participated, along with more than 200 of the participants. So they are interested to be actively participate in this entire event. So by these words, I would like to congratulate and give the best wishes to Mr. Siddharth and entire organizing team, so that we can join tomorrow by the session as per the schedule, which has been delivered and circulated to all the participants. So by these words, once again, Jai Hind to you all and good evening. Thank you so much. Over to Siddharth.
Thank you so much, sir.
Yeah. Okay.
Okay,
so
sir. Then session will be closed. And one more important thing, I say to Miss Sayed Khalida Izar, I will your name shifted to next session, that is
Okay, sir.
tomorrow. Due to some technical issue and your network issue, I will shifted your name tomorrow. I will call you.
Okay, sir.
Okay. Okay. Thank you, sir. Jai Hind.
Yeah,
And
all
close
the
the session.
participants, don't be panic, because whose presentation not accommodated today, it will be accommodated day after, means tomorrow or day after tomorrow also. So if anyone missed their presentation, then you have to directly chat to the Dr. Mr. Siddharth, so that it can be accommodated accordingly as well. Even though Mr. Siddharth will try to accommodate all the presenters and given the time to present their own research. Okay, Siddharth?
Okay, sir. Sure.
Yeah, you have to be in direct conversation to the means young scholars, so that they get chance to present their task. And if selected for the merits, then they also conferred by the medals as well.
Okay, sir. Okay.
Yeah.
Thank you, sir.
So much.
Jai Hind.
Thank you. Jai Hind to you all. So close the session. Okay.
Yes.
Leave the meeting.